All right. Hello, everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. And with me today is my colleague here at NCCAT, Ed Eng. Joining from the Pacific Northwest Cryo-EM Center, PNCC, is Omar Devloku. And from Stanford SLAC Cryo-EM Center, S2C2, who invited today's speaker, is Michael Schmidt. This webinar is a regular event that we host the last Thursday of every month. So please save the date now for our September 29th talk when PNCC is hosting Steve Bonilla from the University of Colorado, who will be talking about using cryo-EM to solve the structures of conformationally dynamic RNAs. And not to be too much of a spoiler, but actually in October, we've got a talk on some small DNAs coming up. So we're going into nucleic acid land for a while, which is really exciting. All of the talks in this series highlight the methods that groups are using to obtain and interpret data they can collect using National Cryo-EM Center resources. And we hope these talks are especially useful for groups just getting started using cryo-EM. Mike is going to introduce today's speaker, Peter Shen, after I let my colleagues from each of the centers give a quick update about the resources we make available at no cost to the research community. And we are recording today's talk. You'll be able to find that recording along with recordings from our past talks on our YouTube page or on the events page at cryoemcenters.org. We will be watching the Q&A box for questions during today's talk, so please use that Q&A button, not the chat button, to send in questions during the talk, and Mike will be moderating those questions for us today. One last bit of business. At the end of the year, we're actually going to host a panel discussion instead of a formal talk as part of this series. And we want to know what people are interested in talking about. So after each of our representatives give their update, I'm going to launch a poll, but I'm going to give everyone now a moment to think what they might be interested in talking and hearing about or discussing in an open panel. Some of the possible topics are specimen preparation, data collection, data processing, logistics of using the national centers. If you are actually here more for science than cryo-EM, that is also okay, and you can let us know. And if you have other ideas or specific things you want to hear about, we want to know what you're interested in. So let us know, and that will help us put together a panel for the end of the year, and that will be more Zoom meeting style, where we'll have all the faces in the room and really have an open forum to answer questions. All right, so with that, I'm going to turn over to our center representatives to give their quick updates. Ed, I will let you start first.
Okay, hello, we're here at New York City for NCCAT. We have 3 main access categories: high-end instrumentation on one of our 4 dedicated Krios instruments, a grid preparation and screening service, and a robust cross-training service. To augment that, we just received our Tundra. Hopefully by next month, we'll have that online for you to cross-train on. And in addition, if you want to stay in New York, we've extended our guest house. So if you want to have an extended stay or room here, please contact the NCCAT user office and we'll work with you. So hope to see you soon. For more information, go to nccat.nysbc.org.
Thank you, Ed. Now I will turn over to Omar.
Thanks, Ed and Christina. I am Omar Devloku, one of the SPOCs, or scientific points of contact, at the Pacific Northwest Cryo-EM Center here in Portland, Oregon. We offer our general access users a seamless workflow from sample prep to 3D reconstruction. And we've also just recently launched a screening access mechanism where we invite users to come on site for a week and work with us hand in hand to go through all of the steps all the way to data collection. We also offer many training opportunities, both remotely and in person, on topics such as sample prep, introduction to serial EM, screening, data collection, and data processing. We've got 4 Crioses, 1 Arctica, and time on a glacios. All of our microscopes are equipped with K3 direct detectors, and a majority are equipped with fringe-free imaging and energy filters. And we're most excited about our latest upgrades, including a Falcon 4i and a cold fig source. I would encourage anyone with questions to reach out, either here through the Q&A or at our website, pncc.labworks.org.
Thank you, Omar. And now turn over to Mike.
Hi, I'm Mike Schmid, and I'm one of the co-directors of the Stanford SLAC cryo-EM Center, or S2C2. Like the other centers, we have three Crioses and time on another Crios and a Talos Arctica. We'll get a Tundra soon. And as the other centers, we have robust programs of data collection, of screening, and of cross-training. And we have all of those opportunities for you. Please see us at cryo-EM.slac.stanford.edu, and please come and see us.
All right, so I'm going to stop sharing, but before I let Mike introduce our speaker, I'm going to launch our poll. And since you all had a moment to think about what it is you'd be most interested in discussing with us, I will give everyone about 30 seconds to chime in with your votes, and you can choose more than one answer. And we're already getting lots of votes, so thank you. I need some Jeopardy music. All right. I'm going to end the poll in about 10 seconds. So if you're on the fence, make a decision. It's now or never. All right. Lots of participation. So thank you everyone for letting us know. I'm going to go ahead and end the poll now. If I ended it before you got a chance to let your opinions be known, shoot us opinions in the chat. But now I will turn back over to Mike to introduce today's speaker.
Hi, it's my pleasure to introduce Dr. Peter Shen from the Department of Biochemistry, University of Utah. And in addition to what he's going to be speaking about today, Lysate-to-grid, lessons learned from cryo-EM of endogenous protein complexes, where you'll learn about how to sort through heterogeneous mixtures of things to pick out what you want. I'd like to make a plug for the cryo-EM curriculum training, the curriculum program, also established by the Transformative Centers for cryo-EM by the NIH. Whatever your style of learning for learning cryo-EM, there are programs established for you to learn in that way. Peter and his colleague Janet Awaza have a very strong visual learning program with animations and visualizations for learning visually. There's a program, if you're more an oral learning person that likes to hear things, there are lectures by Grant Jensen and by Fred Sigworth. There are tactile learning opportunities or kinetic learning opportunities from the cryo-VR people at using VR. Please see that whole program, and it's on cryo-EMcenters.org to show you the curriculum opportunities. And with that, I'll let Peter talk. Thank you.
Thank you, Mike, for that very kind introduction, and especially for the shout out to cryo-EM 101. Hopefully, towards the end of my talk, we'll have time to take a more deep dive into what we hope to achieve through our development of cryo-EM 101. But most of my time today will be spent in discussing our efforts in using cryo-EM to resolve structures of endogenous protein complexes. I have to say, if you were on the poll that Christina sent out, if you did answer the last, if you selected the last option that you're just here for the biology, then unfortunately this talk probably won't be for you. But if you are learning about cryo-EM, you're more of a novice or a newcomer, then hopefully some of the insights that we've learned over the years will be of use to you. This is a rather atypical talk for me to give. Usually I'm, for more cryo-EM centric talks, I spend my efforts in talking about how great the method is. And the method truly is wonderful. But of course, it's most of my time today will be spent on sharing some of the hurdles that we've faced. Now, this audience really needs no introduction to the potential of cryo-EM 101. And it was a fun opportunity for me in preparing for this talk because I had to dig through a lot of old notes that dated back to my years as a postdoc about 10 years ago. And this slide is one that I had shared about 5 years ago when I was on the job market that captured the exciting trajectory of the field at the time. And of course, 2017 was the year that the Nobel Prize in Chemistry was awarded to Jacques Dubochet, Richard Henderson, Joachim Frank for their contributions to cryo-EM. And by that point, the field had matured where near atomic resolution around 3 angstroms was becoming the norm. And in addition to achieving high resolution, cryo-EM also helped the promise of being able to determine multiple structures from a single data set. And this was particularly effective in resolving the compositional heterogeneity. In this example here from one of our papers, a data set of 80,000 particles was used to suss out 15 different states of this particular ribosome complex. And I'll be talking more about this project later. But by now, fast forward 5 years later, this is all pretty old news. But it is interesting to me to reflect on how exciting things were just 5 years ago. And of course, fast forward to our present day. And I think we can all agree that we're very much in the thick of a golden age in the field. We've achieved true, authentic atomic resolution. And there are ongoing developments in image processing that are enabling reconstructions of continuous motions, moving beyond compositional heterogeneity, but also conformational heterogeneity. But of course, these are best case examples, and time will tell if these types of results will become more mainstream. Now, the reality is most samples won't achieve atomic resolution anytime soon, but it doesn't mean that we should stop trying. And there are many samples. For many samples, you can still learn a lot from moderate resolutions. So one of the most powerful advantages with cryo-EM is that you really don't need a lot of material. And so the method is also highly accessible to imaging complexes that are directly isolated from their native sources, and even those with naturally low expression levels. Now, my lab studies complexes that are involved in maintaining protein quality control. And many of these complexes are expressed in low abundance, as low as a few hundred copies per cell, as with this RQC complex. And these are complexes that are challenging to purify through recombinant systems and challenging to reconstitute through in vitro approaches. And so our lab, we reason that cells are already doing the biochemistry, and they're already doing the hard work of putting these complexes together. And so they're already carrying out their various functions in the cell. And so a lot of our efforts are devoted to isolating these complexes directly in the act of them doing work. And so my talk will is structured as follows. I'll start by giving a general overview of how we do our native purifications. We use budding yeast as our favorite model organism. And as many of you can appreciate, being able to purify something is just half the battle when it comes to a cryo-EM project. And so most of my time will be spent on sharing the many struggles we've had with our various projects and how we were able to overcome them. And my hope is that some of the strategies we've come up with would be helpful as you consider the bottlenecks that you might be facing with your own projects. And finally, I'll switch gears by sharing our efforts to teach cryo-EM principles and helping newcomers calibrate their expectations, and thus through our development of this cryo-EM 101 course that Mike alluded to in his intro. So to start off with, just very brief overview of how we isolate native complexes from yeast. The pathways that we're interested in are conserved from yeast to human, and so this legitimizes yeast as a model organism. And they also have the advantage of being genetically tractable, easy to modify their genome. They're inexpensive to work with, have a very high growth throughput. A lot of strong advantages in working with yeast. And so when starting a project, we first identify the protein that we want to co-immune precipitate and its associated gene. And so, all genes will have this general organization. And so what we do is we use PCR to amplify an in-frame tag with its own stop codon, also with its selection marker. And this tag, this sequence is flanked by regions that share 40 to 60 base pairs of homology with a gene of interest. The 5' sequence is identical to the gene of interest all the way up until the stop codon, but not including the stop codon. And then the 3' sequence is identical to the 3' UTR just after the stop codon. And so with standard transformation protocols, the modified gene now expresses an in-frame tag with its new stop codon and then an appropriate selection marker to make sure that the yeast truly have been transformed. And so in this schematic here, this is very generalizable, but here I've indicated that we use 3x flag, and we've dabbled a lot over the years with various tags and have found that 3x flag to give us the best balance between speed and purity. And so just a little bit more about this. Again, so this is highly generalizable, and the advantage here is that you can apply this to virtually any gene of interest. As long as the protein can tolerate a C-terminal tag and it's soluble, then our purification procedures are pretty consistent across many different systems. So after generating the yeast strain, we prepare the cells for lysis using a freezer mill. And this part is fun. I like to show this photo because we drip these cells directly into liquid nitrogen and they freeze on contact. And so we form, these falcon tubes of these BB pellets that we refer to as dipping dots. And others have different protocols that create different shapes like spaghetti noodles or popcorn, but the principle is all the same. We just need a way to freeze cells quickly before they're ground up. And so the instrument we use, this freezer mill, our BBs are filled into these kinds of containers, inside of which is a steel impactor. And so all of the grinding is done under liquid nitrogen, which offers a really strong advantage, compared to other lysis procedures where you don't have to worry about samples overly heating, and this helps to preserve the structural integrity of the components that we're interested in. And so the ground up powder looks something like this. We look for a fluffy and loose consistency, and this becomes the basis of how we do our IPs. And the general procedure is you take this ground up lysate, this powder, and with typical lysate clarification steps with resin binding, washing, and then finally eluting our sample, we have something that's hopefully ready for EM. And the goal of our IP procedures is to have something within 3 hours of thawing the lysate. And just another technical detail, we elute our samples off the resin with a competitive flag peptide. So the end product looks something like this. We typically achieve about 30 microliters, which is enough, plenty of material to run some quality control assays and also to freeze several grids. So we always run these quality control assays, and the two that we always run are, we run our material on a gel just to make sure that our expected components are there. And for a hardcore biochemist, you might look at this gel and be like, well, there's a lot of stuff here. Maybe it's not pure as one might like, but we actually see this as an advantage because for things that we're not interested in, the benefit of cryo-EM is that you just pick around them. But the advantage of working with native materials is that maybe some of these are authentic binding partners or substrates that can't be captured through an in vitro reconstitution method. And so we always run these gels, and our yields are high enough for cryo-EM, but not sufficient to be stained by Kumasi. And so we have our favorite silver stain kit that we use for all of our co-IPs. And then in parallel, we also always run negative stain, TEM, just to make sure that we have good monodispersed particles. Now, that said, these quality control steps aren't exactly a great predictor of how cryo will turn out, as I'm sure many of you can appreciate. And really, we run these steps to weed out the bad preps. So in other words, if the gel and the negative stain look lousy, then we probably won't bother with proceeding to cryo-EM. But on the other hand, if the gel and the negative stain look amazing, then there can still be a long way to go to get good cryo-EM specimens. And so with that, I'll pivot to describing some of these hurdles that we face. So problem number one, first describing an idealistic example. We hope that when we're freezing our grids, you glow discharge the surface, apply the sample, blot and plunge freeze, and ideally your particles are well distributed within the holes of the support film. And of course, this is best case scenario, but life wouldn't be interesting if things worked out so easily. So I started my postdoc 10 years ago, right around the time that cryo-EM was emerging out of the dark ages. And I started working on a ribosome complex. And even then, cryo-EM had a reputation for being a good tool to solving ribosome structures. So being a naive postdoc, I thought that this would be low-hanging fruit. And despite the low copy number of this particular complex, only a few hundred copies per cell, I could purify it directly from yeast lysates. So I freeze grids, go through the typical motions of cryo-EM imaging. And to my dismay, this is a typical image of what I would see. Particles would have a strong preference to stick over carbon and very few particles in the holes. I struggled with this for a really long time, even got desperate enough where I thought, boy, if I just collect enough of these images, maybe I can pick enough particles along the edge of the hole for a 3D reconstruction. I actually tried that and it failed miserably. So these efforts never took me anywhere. So I had to find a way to drive the particles into the hole. So that's problem number one. Well, it turns out that it's well known that ribosomes do not enter holes of cryo-EM grids. And every paper that I was aware of at the time showed that you actually need to freeze grids of ribosomes using ultra-thin carbon. So I thought, okay, this is an easy solution. These are grids that you can just buy, and I like easy solutions. So I collected data of this RQC complex over ultra-thin carbon. You can see that there's particles everywhere. And again, to my dismay, after processing the data, I got 2D classes, but it turned out that there was only a single view of the particle. And so for the cryo-EM novices there, the problem is that with only a single view, you are missing essential information that is needed to complete the 3D reconstruction. And so this leads to really bad 3D reconstruction artifacts, and the resulting reconstruction is not interpretable. Okay, so traditional glow discharging, traditional cryo-EM using a solid support is not the answer. So eventually, I did run out of logical options. But with all the screening that I'd done, the common denominator was that the particles loved to stick to carbon. So could it be that glow discharging the grid was the problem? If particles are sticking to a hydrophilic surface, could making the grid surface hydrophobic instead drive the particles into the hole? So I tested this by washing the grids in chloroform, very hydrophobic solvent. And sure enough, applying the sample to the grid causes the droplet to ball up on the surface. That's the whole reason we glow discharge to begin with. But I figured I could counteract this hydrophobic effect simply by just taking a pipette tip and manually spreading the droplet across the surface of the grid. So the general strategy is to apply a ball of sample on both sides of the grid, and then just manually use a pipette tip to spread the sample across both sides of the surface. The grids are then blotted using traditional procedures, but one important note is that the blot times are really short, usually only between 1 to 2 seconds, because the sample just does not want to stay in the grid. And in hindsight, this really was a crazy idea. I wouldn't be telling you about it if it didn't work. And so this was the result of my efforts here. And this image is the same one that I showed previously, but now by skipping the glow discharge procedure altogether and even making the grids to be more hydrophobic than usual, found that the particles indeed were driven into the holes. It worked beautifully. Now the consistency of the ice could be hit or miss, but the holes with good ice always showed good particles. So this was a huge breakthrough for us to push this ribosome project forward. All right, so that's
Peter?
Yeah.
Hi, we have a question here. Did you try positive glow discharge? And this person said, I have gotten particles into the holes that way when they stuck to the carbon of negative stain grids, but not quantifoils. So
Yeah,
the different styles of glow discharge, I guess.
for sure. Yeah, Jesselyn, I really appreciate this question. I did try polylysine treatment at the time. And this was also probably around the same time that I started dabbling with just skipping the glow discharge grid. I don't have a concrete recollection of how those grids turned out with polylysine treatment. But obviously it didn't work, otherwise I would remember it. But maybe I just couldn't even get good ice with it. But I know that there are several examples that swear by polylysine. And really, this is the spirit of what I think as a field, we should be talking about. What are the anecdotal tricks that people have come up with to get their projects to work? So Jesselyn, I really appreciate that question. But in my hands, I haven't tried glow discharge. I see another question here by Santosh. If the native expression of the protein is very low, yeah, which in my case it is, how would you go about purifying it? So yeah, Santosh, this is a great question. And also I've asked Mike to please interrupt me because I do cover various topics throughout my talk. And so talking about addressing your questions in real time would be more helpful for me. So, I guess very low is all relative, right? And one of the advantages with us working in budding yeast is that we can grow, liters and liters of it without too much effort. But I would say, I'll talk more about this ribosome complex that we're working on. Expression level is about 200 to 300 copies per cell. And typically with about 3 liters of cell growth, we have more than enough material to get good cryo-EM grids. Now, if you're working with, adherent mammalian tissue culture, that becomes much more challenging. But we found that by using a generalizable tag, this has worked for us for samples that are as low as a few hundred copies per cell. So back to our experiences with troubleshooting, this was our first solution in overcoming this issue with particles not entering holes. Now, another common problem that we faced in various projects is that of particles adopting preferred orientations. I think those of you with more cryo-EM experience, at some point you are going to encounter this issue. And so this is a different project now. This is one of a AAA ATPase that really is a cornerstone of my lab. It's called CDC48. And here, thankfully, we didn't have an issue of particles not entering holes, but we instead had a problem where particles adopted very strong preferred orientations. See from the 2D class averages and the resulting 3D reconstruction that the top view was very reasonable, but there was just a complete absence of side views. And so the artifact here, you can see that on the side view, it's very streaky. And so there was very restricted range of views using our traditional grids and very smeary on the side view. So we tried a few known tricks, including adding detergent, using ultra-thin carbon. But for this particular project, those other tricks didn't seem to solve the problem. But it was also around this time that gold grids were becoming popular and also more accessible. You know, you could just buy them from the company. So for reasons that are still unknown to me, using gold grids was enough to give us just enough side views over here to help us achieve an overall resolution of 3.7 angstroms of this particular complex. So thankfully, in this case, the solution turned out to be very straightforward. We only needed to switch the type of grid that we were using. And so nowadays in my lab, I do encourage people to always use a variety of grid types when they're optimizing their grids. Of course, the major advantage to gold is lower beam-induced motion, but this also had the side benefit, this really important benefit of giving distinct views, again, for reasons that I can't explain why. Now, also around the same time, we were studying a different AAA TPAs called VPS4. This is a project in collaboration with my colleague, Chris Hill, in our department. And so again, using normal Vitrobot-based freezing on quantifoil grids, we saw a similar effect where we had an enrichment of top views. We also had some side views, and so we were able to get a reasonable overall 3D reconstruction, but you can see here in the orientation distribution plot that there's still a clustering, strong enrichment of views in one direction. So again, this is only about 4.5 angstroms. And with what we know now, particles have a high tendency to adopt preferred orientations because they love to stick to the hydrophilic air-water interface. And so we became aware that the spotted on device at the New York Structural Biology Center could be used to essentially outrun the particle interactions with this interface. And this could help resolve the orientation bias problem. And so with the support of Bridget and Clint at NYSBC and with the direct help from Venkat Dandy, he helped us to prepare these grids using spotted on. The images were just absolutely beautiful. Now, in this case, though, what we were expecting was a more complete set of views in this reconstruction from the spotted on grid. But instead, what we got was a different set of preferred views. With the Vitrobot, we got an enrichment of top views. And with spotted on, we got an enrichment of side views. And so each of these data sets independently produced 4.5 angstrom structures. So of course, with these views being complementary between these data sets, it made sense to combine them into a single reconstruction. So even though spotted on itself wasn't the answer, the advantage of having different views recovered using different grid freezing methods enabled us to push this overall resolution from 4.5 angstroms to better than 3.5 angstroms. And so this was something that spotted on really helped us to overcome this hurdle of preferred orientations. Now, the last hurdle that I want to share with you, and probably one that will take the most time, is one that's that we face all the time with our interest in multi-component complexes. So going back to the ribosome project, I've told you that we can isolate these native complexes directly from yeast lysates, and that we've worked out ways to drive the particles into the holes of these hydrophobic grids. But sadly, this was not the last hurdle. We collected a huge data set at the time, something like 2,000 micrographs, which at the time was, took a heroic effort, and semi-manually picked hundreds of thousands of particles, which, remind you, we didn't have really robust auto pickers at the time. But the point is that it took several weeks of data processing just to find out that our 3D reconstruction was that of an empty ribosome. So even though in our gel, we expected to see an enrichment of these bound components of the RQC to these ribosomes, in the end, our reconstruction only showed evidence of the ribosome itself. There was no evidence of the bound RQC factors that we were interested in. So what happened? And the most logical explanation is that at some point between purifying the sample and freezing the grids, the complex had fallen apart. So nowadays in our lab, we've routinely used a number of strategies to counteract this effect. And one such approach is by designing informed mutations into our proteins of interest. So just to get a little bit more into this biological pathway, so why did the complex fall apart? We reasoned that one explanation is that the complex was still active during the course of purification. And so the job of this ribosome complex that we're interested in is to remove these faulty products of translation. And so there are multiple proteins that come on, and this pathway culminates in the recruitment of the CDC48 segregase, in which it pulls out a ubiquitinated substrate directly out of the ribosome. And so we asked if we could essentially trap this complex in an intermediate state, and that if we could do so by blocking the recruitment of CDC48. And so we did this by engineering a catalytic dead mutation of the E3 ubiquitin ligase that is responsible for ubiquitinating the stalled nascent chain. And so this is the principle behind how we wanted to trap our complex of interest. And after generating this mutation, we did the quality control, made sure that our complex was still recoverable by co-IP, which it was. And then this time we used 3D reconstruction and 3D classification. And even with all of these efforts, collected a data set of about 80,000 particles. And we did a lot of classification with it, and the most complete structure was reconstructed with only 5,000 out of the 8,000 particles. That's something like 6% or something like that. And so the overall resolution wasn't that impressive, only about 10 angstroms in this case, but the structure still enabled us to tell a story of how this RQC complex handles stalled ribosomes. So 6% is better than 0%. So in this case, generating a specific mutation to trap the complex in a desired state was needed to help us to push this project forward. We've also used other methods to trap particles in their desired states. Going back to this AAA ATPase complex, when we do these native purifications, over the course of the purification, by the time we freeze grids and solve the structure, we recover these complexes in an inactive state. And so in the final reconstruction, we can see beautiful, these are ATPases, and the nucleotide binding site, we see beautiful density for ADP. But of course, what we're really interested in is to pull out these complexes while they're still in the act of doing work. And so we reasoned that, well, maybe we can accomplish this by using a non-hydrolyzable ATP analog. And I would say that for many of our projects, this ADP beryllium fluoride analog is our secret sauce. This was the nucleotide that was determined by Chris Hill's lab for this other triple ATPase called VPS4. And in our hands, this will also worked beautifully for CDC48. The principle being that by including this analog in the lysates during the course of purification, now the ATP, now the nucleotide can't be hydrolyzed. And so any complex that was in the act of doing work at the time would essentially be trapped in that state. And that's exactly how it turned out for this particular complex, where we were able to resolve 3.7 angstrom structure of this protein unfoldase in the act of unfolding some substrate that was within the yeast cells at the time. And we were at high enough resolution where we can model in an ATP-like density. There's ADP beryllium fluoride, but it's ATP-like in our final reconstruction. So with mutations, with using the right small molecule analog, these are a couple of strategies that we routinely use in trapping our structural targets in a desired state. And so the last solution that I want to share with you is our use of chemical cross-linking in our native purifications just before plunge freezing. By 2018, 2019-ish, important studies by Alex Noble at NYSBC and Werner Kohlbrandt's group confirmed these long-held suspicions that particle interactions with the air-water interface was a major driving force behind common problems associated with cryo-EM, including preferred orientations and particle denaturation. And so we reasoned that a mild amount of cross-linking could provide some level of protection to these particles to keep them intact during grid preparation. Just saw a chat pop up.
Yeah.
Rashmi asks, are these analogs added to the lysate? They are. So just backtracking here. Yeah, these analogs are added to the lysate, and they're also included in every wash step. So as soon as, by the point they're added to the lysate, basically all existing nucleotides during the course of purification will be washed away, but the analog would persist. And this is an important point because we want the complex to hold on to their substrate during the course of purification. Thanks for that question. All right, so particles have this high tendency to, bad things happen when they interact with the air-water interface. And so could we protect them with some level of chemical cross-linking? And so our favorite cross-linker is glutaraldehyde, very general cross-linker. Cross-links primary amines, but with variable lengths. And so in order to get the right amount, we do a titration with increasing amounts of glutaraldehyde. And we strive to achieve this Goldilocks zone where a mass shifted material is enriched towards the top of the gel, but also some uncross-linked material remains. So probably this rightmost lane is probably over cross-linked, and then this lane right next to the uncross-linked lane is probably insufficiently cross-linked. And so you find some balance. We always do the negative stain to make sure that that monodispersed particles are still there. And so one variable we use is different concentrations of the glutaraldehyde cross-linker. Another point of optimization is time. And so we typically do these reactions on ice. And again, trying to find the sweet spot of finding a balance between cross-linked and uncross-linked material. And so this is an example from our recent publication. Well, I guess a few years ago now, still recent in my mind. But this, here in the right two-most lanes, you can compare the cross-linked lane on the rightmost lane with the uncross-linked lane. And this is generally what we strive to achieve, a good mix between cross-linked material, this mass shifted stuff on the top, and then still seeing the original bands for the components of interest. So again, this does require a bit of optimization because every sample is going to be different. But as a starting point, if this is something that you're interested in, we would recommend about 0.01% glutaraldehyde for 10 minutes. And use that as a starting point to make sure that, use the negative stain to validate that your particles haven't been overly cross-linked.
Peter, I have a question then. Is the concentration of the protein itself a variable, or you just simply want it not to aggregate by cross-linking it to another?
Exactly. Yeah, no, it's a really important point. The level of cross-linking that we're striving for is the with an intra-particle cross-linking, right? And of course, if you give the reaction enough time, you'll get inter-particle cross-linking. You bring up a good question, though. Something that I haven't alluded to is the concentration of our preps. And concentration is going to vary with every sample, but with these native purifications that we do, we typically get on the order of 100 to 1,000 nanomolar concentration, so 0.1 to 1 micromolar. So relatively low amounts when it comes to a structural biology standpoint, but we're able to determine some cool cryo-EM structures despite these low concentrations. And so yes, your cross-linking conditions then will be dependent on the concentration of your prep. And so if you're working with some recombinantly purified protein, you got several mgs per mil of the stuff, then sure, those cross-linking conditions will be different. And so that's why, I show you these couple of examples of the titrations that we do. And we, every time we have a new project, we do have to figure out, all right, what is a proper level of cross-linking for, this particular project.
So we have a couple of other questions in the Q&A. Is this similar to graphics, the gradient fixation? Do you need to ultracentrifuge the glutaraldehyde treated species?
Good question, Carl. Thank you. So graphics is basically a very clean and refined version of chemical cross-linking, because now you're also separating your contents out on an ultracentrifuge too. We don't run graphics. We don't run graphics because introducing a multi-hour ultracentrifugation step kind of defeats the purpose of us trying to go through these ultra-fast purification, these fast purifications that we try to achieve within a few hours. And so we found that just adding the cross-linker to the elutions is sufficient to preserve their structure. So this gets at Nancy's follow-up question here. Does the cross-linking happen in lysate or purified protein? We do it with purified protein. Again, the reason is really to protect particles when just before they're blotted and plunge frozen, right? They're probably fine and happy in solution for for a certain period of time, but it's really that act of blotting and particles colliding with the air-water interface that causes particles to break up. And so by adding the cross-linker just before blotting and plunge freezing, this provides some level of stability, which I'll show in the next slide. And do I have a favorite quencher for cross-linking? Yeah, we just use tris. So another important note, thanks for the question. We do all of our IPs, our purification is a heap-based buffer. Because if we were to do it in tris, then tris with all of its primary amines would make that sample inaccessible to glutaraldehyde-based cross-linking. So we do our purifications in heaps, and then if we cross-link, we cross-link in glutaraldehyde and quench in tris. And let's see, Carl, follow-up. Do I need to do dialysis after treating to remove excess glutaraldehyde? No, we don't remove the excess glutaraldehyde because we quench from excess tris. I think the concentrations come out to be about 1 millimolar final concentration of glutaraldehyde and about between 5 to 10 millimolar of tris. So we add excess tris, but 5 to 10 millimolar we feel is tolerable. What happens if the cross-linking gives a complex which is not real? I mean, I think this is the age-old question, of the age-old concern with introducing any form of fixation or cross-linking. And I don't have a really satisfying answer to your question, but I'll just say that just let the structures speak for themselves. It's always a specter that's looming in the back of my mind, but that's also a reason why we strive to, when we cross-link, to achieve a balance between uncross-linked and cross-linked material. Yeah, it's an important question. All right, so let me show you some results of our efforts using cross-linking. So as I alluded to earlier, by removing the catalytic domain of this LTN1 E3 ubiquitin ligase, and after extensive 3D classification, we were able to achieve a 10 angstrom structure from about 6% of the particles following 3D classification. After cross-linking, we were able to achieve pretty much a very similar structure, but now with a highly enriched population of intact particles. And in this case, we were even able to observe new density that we hadn't detected before. Yeah. So I would say if we use this non-cross-linked reconstruction as a positive control, at least there's a lot of similarities here. And so in this case, I would say the structure speaks for itself, that we haven't introduced any concerning artifacts. Now, this glutaraldehyde-based cross-linking was also instrumental in our work with AAA ATPases. And in collaboration with Chris Hill's lab with VPS4, this particular hexameric complex in solution is a very weak hexamer. The equilibrium between shifts skews very highly towards the monomer or dimer of this particular protein. And so in order to stabilize or promote the hexamerization of this complex. The Hill Lab has fused this expression construct to a scaffold called HCP1. Details aren't important here. Suffice it to say, though, that when we imaged the non-cross-linked material, even at very low thresholds, like we saw very weak densities for VPS4. But with the same glutaraldehyde approach that we use, this was sufficient to stabilize VPS4 in its hexameric state. And so I would say that we're at a point now where every cryo-EM project that we embark on, we always include some level of glutaraldehyde-based cross-linking. I understand the caveats that it's not perfect, but I do see it as a necessary evil as long as we're using these blot-based plunge freezers where particles are constantly getting shredded by the air-water interface. So just to recap, some of the strategies that I've shared with you today described our efforts to overcome issues with particle partitioning, with preferred orientations, and complexes falling apart. But in reality, this really just barely scratches the surface. When it comes to a number of things that one can try to tackle these common problems. And so here's a list of strategies and tools that I put together that has been described throughout the literature. And even this list is not really comprehensive. And you can imagine that the combinatorial possibilities of trying a matrix of different strategies will quickly get out of control. So as awesome as cryo-EM is, it still has the major disadvantage that the throughput of screening samples is extremely slow. We don't have anything like a crystal tray to screen a bunch of conditions. Each grid has to be examined individually. And I think that's really just the tough reality of where we are currently as a field. And all of the latest bells and whistles and microscope hardware and image processing software won't be helpful if we're bottlenecked by screening. Now, my intention is not to cast pessimism. I think the future is very bright. But if I were to distill my thoughts into a single statement, it would be that there is no magic bullet to making the perfect good. And trial and error will be necessary, but we can go about it in an informed way. If the issues are that your particles aren't entering the holes, then at least that means that there's a manageable number of things to try. Same thing with orientation bias, what complexes falling apart. Now, hopefully your samples don't have all of these issues, but even if so, chances are someone has figured out a way to resolve a similar issue. And so my recommendation would be to talk to experts at the national centers, because they've probably seen more different types of samples than anyone else. And they've probably been able to come up with solutions on their own, or are knowledgeable of the literature where they can offer meaningful suggestions to overcome these bottlenecks.
No pressure.
Yeah, that's right, Mike. Hey, that's your job, man. No, but I do think though, the NIH recognizes that this is a big issue, right? And so that's why they've offered supplements to the national centers dedicated to screening specimens. I think it's wonderful that the national centers have so many microscopes to collect data. You guys have all the latest hardware, but I don't want to speak for you, but my assumption will be that, screening grids is taking a lot of your time and remains a huge bottleneck.
Yeah, and one of the possible avenues for doing this, now that we have this menu, pick one from column A and one from column B, et cetera, et cetera. There are programs which are design of experiment programs that sparsely sample, kind of like crystal screening grids, crystal screening kits that they used to have, that sparsely sample this matrix of all the different possibilities and analyze the results to zero in on what is working based on combinatorial mixtures of these different choices. and that's one of the possibilities that that it involves machine learning or analysis of these results of screening. Of course, you still have to do the screening, but smart design of these kind of experiments for navigating that full matrix is something that I know some people have tried.
That's great, Mike. I wasn't aware of that, but I'm sure that there are a lot of smart people out there that recognize that this is a huge problem. And I truly think that there are more floodgates ready to be opened when people can overcome these issues in a very general way. Could there be a magic bullet that maybe not a one-size-fits-all solution, but at least something that can resolve a lot of these common issues that too many people are experiencing? So I want to be sensitive to time, and I've do want to just take a couple minutes to introduce cryo-EM 101. One of the motivations for me and Janet Iwasa to develop cryo-EM 101 is recognizing that it's still the wild, wild west when it comes to people coming up with their own tricks and solutions to making the perfect cryo-EM sample. And so we teamed up to develop this website, and I won't talk about it extensively because all of you can just log on to it and see for yourself. But with Janet's expertise as in molecular visualization, she's really put in a lot of effort to develop these really visually effective tools to teach principles using illustrations, animations, videos, and interactivity. But the one thing I do want to just bring to everyone's expectations is that we hope that this becomes a resource where people, especially newcomers, can calibrate expectations when it comes to them delving into their own cryo-EM projects. I think the field itself has perhaps done itself a bit of a disservice in, giving, especially like news outlets and giving an impression that, cryo-EM, it's going to displace X-ray crystallography. It's going to, solve everyone's problems. And so I think having a resource to help people calibrate expectations is really, really important, and it's one of the major goals with cryo-EM 101. So I'm just going to fast forward here towards the end of my slides to show you, just to describe one thing that I hope we can achieve through a community effort. For me, one of the best ways, one of the fastest ways for me to learn is actually by making mistakes and learning through bad examples. Too often are we reading papers and our expectations are being miscalibrated because we see these examples of beautiful representative micrographs. And so when we see that, our own images don't look like that, what are we doing wrong? And the reality is that, a very small percentage of the data sets that we collect actually end up contributing to a high resolution structure. And more often than not, our images might look something like this. And there currently isn't a great resource where, bad images can be used as a learning tool. And so in cryo-EM 101, we have a section in chapter 3 here where we have a gallery of bad examples. And so we have good examples, of course, but this is a home where we hope people can learn, what do bad examples look like? So I'm at time, and so I'm going to end on this note, a plea, if you will, because I'm joined here today by a lot of cryo-EM experts and a lot of cryo-EM newcomers. When you're collecting your own data, chances are you're going to have a bunch of lousy images. Send your lousy images to me, and we'll find a home for it as a learning resource on cryo-EM 101. So with that, just want to acknowledge the major players involved that have guided me throughout the years and who are continuing to work with me now. My lab, I'm grateful for them for their efforts in bearing with me. And as a postdoc, worked with Adam Frost, Owen Brandon, and Jonathan Weissman on the RQC complex, continuing to work with Chris on AAA ATP ACEs, and Han was a former postdoc in his lab, and our ongoing efforts to develop cryo-EM 101 and also now cryo-ET 101 are in partnership with Janet Iwasa and Julia Brash, and my funding sources are on this slide. appreciate everyone for their attendance. I saw a number of questions pop up, so I'll take some time to answer them now.
Yeah, thank you so much, Peter. I just want to, for people headed off to another meeting, thanks for joining us. I really love how you point out there are problems to solve and we can solve them better as a community. So I'm extremely happy that we're all here in the room together, admitting everything is not as easy as it looks when you just read the method section of a paper. So yeah, we can stick around to answer those last couple of questions, and we're still recording, so those will be included in the recording if people need to pop off. And yeah, if you're willing to stick around another 5 minutes, we'll let you field those questions. So go ahead.
Thank you, Christina. All right. First question here. I have a small related problem. Purified a protein. SEC looks good, but on negative stain, protein is aggregating and forming different sizes or shapes. Suggestions on how to deal with it. I think, this is the type of question to punt to the experts at the national centers, right? Probably, these are the types of questions that you are probably dealing with on a near daily basis. And so I don't have a magic bullet answer to this question. Again, every protein, every sample is going to have its own characteristics. But when it comes to aggregation, there are certain things to try. Is your concentration too high? That would be an easy problem to fix. Forming different sizes and shapes, I think, unless, other experts have a chance to look at the data, it's hard to provide a blanket statement that will address this particular question.
It's an interesting dichotomy. You're worried about things falling apart. Other people worry about things sticking together, and doing the opposite of what you're doing, that is cross-linking, perhaps interrupting interactions. Perhaps detergent or something that interrupts interactions. Maybe difference in salt. There are a lot of electrostatic interactions that would be shielded if you added higher salt. So things like that to try when you think about, okay, things stick together. Well, let's go through the objects that the things that make things not stick together.
Yeah, and so the first thing I have on my list here is buffer composition. I mean, this is something that every biochemist will include on their list of things to adjust. And yeah, it's as a structural biologist, I think we can all appreciate that biochemistry will always be king. Appreciate the comment on cryo-EM 101, and then the question I see here from Natalie: have we tried adding the analog later to the purified protein with similar results? We always add the analog during the course of purification. The reason being is that we want to retain the complexes while they're still bound to substrate. And so the reason we don't add it after is that we would be concerned that during the course of purification, substrates could be released, things could fall apart. And so adding the analog later might be too late. Though I don't recall if we've actually tried it. Have we tried other nucleotide analogs like AMP-PMP? We haven't tried AMP-PMP. Other papers studying similar types of structures have used ATP gamma S. The reason why ADP beryllium fluoride is our secret sauce really goes back to beautiful chemistry done by Chris Hill's lab several years ago now, where they saw that for this particular family of proteins, these AAA ATPases, that ADP metal fluorides, so beryllium fluoride or aluminum fluoride, were the best when it comes to substrate interactions. And I don't believe that they tried AMP-PMP in their case and it just didn't work. But again, I think sure, there are a lot of other structures that have been solved with AMP-PMP, but just in our hands, we haven't explored that. Membrane protein complexes. This is something that I intentionally didn't describe because membrane proteins are an entire different beast on their own. I will say that we have integrated our 3X flag tag on membrane proteins and that the purifications look good. And by negative stain, silver stain, the major difference is, the inclusion of detergent at some point during the purification. But our major bottlenecks have always been at the cryo-EM level, because the inclusion of detergents is just a deal breaker in our hands, especially considering the typical low yields of native complexes. So I admittedly am just not an expert when it comes to membrane protein biochemistry. So I don't have any recommendations for you, Tomas. Favorite tags to aid picking and contrast. Yeah, if I were more comprehensive, James, I would add that suggestion to this list. In our hands, we haven't tried inclusions of fabs or Lego bodies, anything of that nature. Typically, those appendages would be really helpful if you're dealing with a really small protein, but the need hasn't arisen in our hands for our projects, so I can't comment on that, James. Is there going to be cryo-ET 101? Why don't you log on cryo-EM101.org right now, and that will answer your question. Short answer is there is going to be cryo-ET 101, and the first chapter is already posted. Rashmi asks, any idea from experts? Well, good NAD plus analog. Can't help you there, but I appreciate you using this forum as a way to get some suggestions. That's just not my field.
Anyone else listening could answer or contribute to the Q&A that way, but that's a good question.
All right, I think that brings us to the last of the questions I see there. So thanks again, Peter, so much. That was absolutely wonderful. It's good to know we're all in the same company when it comes to making good grids.
That's great. Thank you, everyone. I appreciate this
All right.
opportunity.